Hello and welcome back to this channel. Remember I said I was going to come back and explain all the biology Waec practical specimen for the year 2024. And this exam is fast approaching, actually next week Friday being the 24th of May. So I'm here to explain the first specimen, and this video is particularly about explaining the first specimen, which is what the gill of a fish. Now, one thing you must know is this. Whenever you want to answer Waec practical questions, if you have something like the gill, look for another thing that serves the same function, still part of the specimen given to you. And another thing that serves the same function, first, what is the function of the gill? We know it's for respiration. Now, another thing that serves the same function, just like the gill, which is part of the specimen provided for this year's biology practical, is specimen C, which is what? The lungs of the domestic fowl, because the lungs is one organ that is involved in respiration. So therefore, you have to know some differences between specimen A, the gill of the fish, and specimen C, the lungs. You have to know some differences. You have to note some similarities. You also have to note some observable features of specimen what A, which is the gill of a fish. So that is what I'm here to explain. Now, with these tips, I bet you must definitely see some possible questions from all that I'm going to talk about relating to the gill of a fish, specimen A, and specimen C, the lungs of a domestic fowl. So let's dive right in. Now I'm going to start with the first point I want you to note. Note this, the gill of a fish. The gill of a fish is also sometimes called the branchia, gill or branchia. Now, the gill of a fish it can be seen in any. If they ask you first, they can also ask you questions like, where is specimen A found? You could say it is found in a fish. Particularly, you could use the word it is found in a bony fish, because the bony fish has a gill that is a bit easily accessible. Do you understand? You can easily access it and you can easily see it around. And if they ask you, for example, you could say an example is a tilapia fish is a bony fish, or a catfish is a bony fish. Then the next thing they may ask you list some observable features of a specimen A, which is the gill of a fish. What are these observable features? Features you can see. Yes, you know that this thing exists, but you cannot see it. For example, if they ask you observable features of probably the gill of a fish, what can you see at that time? Number one, it is moist. Remember that Waec has asked the schools to present a freshly procured gill, meaning that gill must still be moist because it was just freshly brought out from the body of the fish. Or maybe they brought it out and they kept it in the fridge, so it must be moist, meaning that it has wet or some kind of water feeling or water on it. So when you see it, you notice that you can see water. When you touch it, your hand will be a bit wet. So that is number one point. It is observable there. You can see it. Not something that you have to open before you see it. That is not observable. Observable is something that you must be able to see. So the first point, observable features of a gill of a fish is number one, it is moist. Then number two, it is it has we can use the word, you always put it presence of, if the thing is there. So let's say presence of the what? The gill filament. There's a gill filament, a comb-like structure that has lines on the gills, the gill filament. The other one you could also say is presence of what we call the gill arc. These are observable features. Then you could also use another observable features is that it has a large yes, you could use that one because it's observable that it has a large surface oxygen gas which comes in, and of course carbon dioxide which goes out. So it therefore tells you that the gill of a fish has these features. Number one, it is moist. Number two, it has large surface area to increase the rate of diffusion. Number three, I didn't say this, but you should add it. It is brown, reddish in color, or you could say it is reddish brown in color. That's another thing, you can see the color. Then number four, it has the presence of the gill filament, which is a comb-like structure. Then another point is presence of the gill arc. The gill arc is gill, and then arc is spelled ARCH. Now the gill arc is that part that joins the filament with the gill raca. So the gill raca enters inside, like you can see in the second diagram. So the gill arc is that part, the gill arc is the part that holds the gill filament and the gill raca. So let's go to another point I want you to note, which is some observable similarities between specimen A and specimen C. Remember that specimen A is the gill, then specimen C is the lungs. So what are those things that you can observe between both of them? Well, now, both of them have a pink/reddish color or dark red color. Then both of them have, you can say both have, you can say both of them are red or they have a pink or red color. Then the next one is that both of them have a moist surface. The lungs must also always be what moist, you get, because it was just taken out of the body of the domestic fowl, and it must always be moist because where it is found inside the body, it is moist. Why is it moist? You can add that extra point. It is moist to what bring about the diffusion of gases, or bring about it's moist so that it can dissolve gases. Do you understand? So this is another point. First point is both of them are red or dark red in color, or you can say reddish in dark red. Sometimes are red. Do you understand? So this is another point. First point is both of them are red or dark red in color, or you can say reddish in dark red. Sometimes you can use the word pink. Don't use the word pink. Dark red is the perfect word in color. Then the next one is that both of them have what moist surface. Then both of them are what you would say they are highly vascularized. Yes, that means they have a network of capillaries and veins that constantly supplies blood to both the gill filaments and to both the gill and the lungs. But because you can't say these capillaries because they actually observable features, but they don't said observable because they asked you observable similarities. But I didn't mean they said similarities without the word observable. Yes, you could put down this point, which is that both have a network of what capillaries. That means they are highly vascularized. Then another thing you could say is that both have a large surface area. It is seen there. So I've given you 3 points under some observable similarities between gill of a fish and domestic fowl. The first similarity is that both of them are reddish, dark red in color. Both of them. The second one, both of them are moist, or they have a moist surface. The next one, they have a large surface area. Then the next one is you could also add that both of them have a thin wall membrane. Thin wall membrane. That's thin, THIN, then walled, like WALLED. That's WALLED, like a wall of a building. Yes. So both of them have a thin walled membrane. Then let us go to another point. You could ask you the similarities between gill of a fish and the lungs of a domestic fowl. Now, what are their differences? Now, you have to rule or answer this question in a tabular, or what we refer to as a column form, where you have a column. On the first column is for what? Gill. Second one is for lungs. You rule the line to differentiate. It is best to answer this way so they can easily understand your point and give you your full mark. Remember that practical carries 40 marks, then theory and OBJ carries 30 marks each. So for this practical, you need to do your best. So let's go back to the difference between specimen A, which is a gill, and specimen C, which is the lungs of the domestic fowl. Now under the gill, you write the presence of gill racas. Racas is spelled RAKERS. Gill racas. Then under the lungs, you write absence of gill racas, or gill racas are absent. Then the next point, you have the gill arc is present. Under the lungs, gill arc is what? Absent. Arc is spelled ARCH. I think you can see that in the diagram. Then the next point, number 3, is the gill lamella is present. Then under the lungs, the gill lamella is what? Absent. Lamella is LAMELLE. Remember that the lamella is the point where the gaseous exchange occurs. In this lamella is inside the what? Filament. So in case you are asked, where does the gaseous exchange occur in the gill? It occurs in the lamella, which is found in the what? In between the gill filaments. Then the next one you could say is under the gill, pleural membrane is absent. Pleural membrane is pleural is spelled PLEURAL. Then membrane is what's absent. Then under the lungs, pleural membrane is what's present. This is a membrane that surrounds the what? The lungs, the thoracic cavity of the what? Lungs. So the next point under the gill is that the pleural cavity is what? Absent. The first one was pleural membrane. This one is pleural what cavity is absent under the gill. What is present under the lungs. Do you understand? So at least I've given you like 4 points or 4 differences between the gill of a fish and the um, lungs of a domestic fowl. Then you should also learn the drawing of the gill of a fish. And when you draw, that's like I said in my earlier video, always try to label up or down, and always try to give the title of your diagram either up or down after the drawing, either before the drawing or under the drawing. Then your labeling should come on your right hand side, all labeling on one side. And the lines that show what you are labeling should all start from one point and all end on the same point. So you don't have some long lines and some short lines. Then you label all on one side, either on the right hand or all on the left hand side. Don't label some on the left and some on the right. That reduces your mark. Another thing is title. You should title your diagram of the words. It should not be diagram of specimen A. No, it should be diagram of the gill of a word fish. You understand? Then your size. Remember they always tell you make a well labeled diagram 8 to 10 centimeter, or sometimes make a well labeled diagram 10 to 12 cm. Please, if you are using that scale, you should always know that 10 is the minimum. Meaning, if you put your ruler from 0 cm to 10 cm, you can use that, or you keep your ruler and calibrate or measure from 0 cm to 11 is okay, or from 0 cm to 12 is okay. But when you get to 12, you shouldn't have anything. Your drawing should not exceed any measurement of maybe 12.1, because you have gone outside your the diagram or the range or the size they gave you to use in your drawing. Remember that's remember also that range or that size given to you is what you're going to use to obtain your magnification, which every drawing should have. Magnification is calculated as image size over object size. Please, image is how do you get your image? Your image is what you draw in your paper, while your object is what the specimen that has been placed on the table or on the stage before you. So when you go there, you use your ruler to measure what has been placed before you on the stage. You measure from 0 to whatever cm you get, 0 cm to whatever the thing you got the measurement. That is your object size. Then you go back to your book. If you are using seats, probably they told you using a scale of 8 to 10 cm. If you are using 10 cm, that is your what object size. If you are using 8 cm, that is your what object size. So you use that. If for example, if you measure the object, that is whatever that was mounted as the gill before you, and you got something like 4 cm, and then you went back and you noted that what you want to use to draw is 8 cm, not 10. Now what will happen is that your own object size will now be what 8 cm. Sorry, your image size will be what 8 cm. While your object size is what you measured, the real gill that is on the stage that you measured and you got 4 cm. So since the formula is image size over object size, it means that your image, which is the one you draw on your paper or in your book rather, is what 8. Then the one you measure, which is object size, is 4. So you divide 8 by 4, which is going to give you what 2. So your magnification will now be after your title, diagram of this of the gill of a fish. At the end of that full stop, when you finish giving your title, on the same line, you now put mg times what 2, because you got 2. 8 divided by 4 is 2. Why are you putting x? Put that x before mg means magnification times. We put the symbol of x, like the X alphabet. You put it before you put the 2. Do you understand? So because that 2 means that whatever you draw in that book, whatever you drew as your diagram of the fish, it shows that diagram times 2 of it is what you have as what is mounted as a specimen, the real gill on the fish. So it means that you have more increased or amplified the size of that that specimen. You've reduced it on your book, but times 2 of it will give you what is on the word stage. Do you understand? Don't forget your magnification. Always use HB pencils to draw because they are not thick or dark. Then always you can use 2B to label, or you can also use the same HB to label. So these things are what Waec look for in drawings. They look for title. There are 5 things they look for: title, magnification now rather. They look for title, they look for the size of the diagram to make sure that you use that range that was given to you, 8 to 10 cm or 10 to 12. You do not draw lower than 8, which is 7.9. Anything from 7.9 is lower than 8, or you do not draw more than the highest measurement, which is 10. For example, if it's 8 to 10 cm, so you did not draw something that was 10.1. So they use title, they use size, they use clarity of line. Your line should be clear. That's why I say use HB pencil. That pencil that is yellow, or you can have the one that is yellow and black, but don't use that one that the technical drawing students use, which is dark. I think that one that is blue with black lines is too dark for biological drawings, and it will not give you your mark on that clarity of line. Also, neatness of labeling is another thing. Spell correctly, label words clearly so that the examiner or the person marking can see it. Do you understand? So with this, you get your full mark. And with this, I've come to the end of the. Okay, sorry if there's another point you need to talk about on the gill. I've spoken about the gill, that is an organ of respiration. Okay, there's another thing I need to see. I know that I only gave the difference between the gill and the lungs and the similarities. Then I also need to point out some things you need to know about the gill, which is where the gill is located in the fish. It is located behind the eyes, and the gill is located at is inside the gill cavity, where the upper column cavity. The gill is covered by a flap we call the upper column or upper collar when you're talking about one. But we're talking about the both sides where the gill is located on the head. Behind the eyes of the fish is called the upper column cavity. So the upper column is that flap that covers the gill. When you open it, you see the gill, but when it's closed, you don't see the gill. So the upper column is another name for it is called the gill cover. It covers the gill. Then it is the inside the gill. How does this gill work? The fish opens the mouth. Water from the environment gushes into the mouth of the fish, and then passes through the gill filament. Remember that the filament will just flap and then exchange gases, because the inside this filament, in between the filament, you have the lamella. Lamella is where we have network of blood capillaries. So that is where gaseous exchange occurs, whereby oxygen comes into the gills and carbon dioxide diffuses out. Then the gill raker, what does it do? The gill raker rather has something it does. It filters food that comes in with the water, food like tiny larvae or all these insects that the fish might feed on. You understand? It filters it and eats. It doesn't have anything to do with respiration, the gill raker. Then the gill arc is that point that connects the gill raker with the gill filament and the lamella. So this, I'm sure I've explained how the gill works. The point where gaseous exchange occurs, which is at the lamella found in the gill filament. And then I've said some things about the location of the gill and the structure of the gill. So with this, I come to the end of the video on the first specimen for the 2024 biology practical Waec examination, which is the gill of a fish. Now I hope you stay tuned and watch the next video on specimen B, which is nothing more but the liver of a domestic fowl together with the gallbladder attached to it. See you in my next video and stay tuned. Bye.